So, hello everyone. Today we're gonna know how to do cell passaging. This is a basic type, and this is mostly used for the cancer cells. And there are like some kind of cells which are sensitive to trypsin, and they use EDTA, and they also spin down the cells, and the steps are basically a bit different. But this is the basic cell passaging and the simple way to passage your cells. Let's get on to the process. So initially we have to open the window of the laminar airflow and on the laminar airflow and the light, and you have to clean all the working surface with 70% of ethyl alcohol before bringing the things into the hood. So after cleaning the hood, bring the flask that you wanna passage and bring the trypsin, PBS, media, and the pipettes and pipette gun and the tips and the pipette tubes for pipetting gun and the new flask for the passaging and a waste container. So after bringing everything into the hood, remove the media that is present in the flask by making an angle of 45 degrees, and with the help of pipette gun, suck all the media from the flask and dispose into a waste container. And then after, rinse the whole surface area with PBS. You can take around 2 to 3 ml of PBS and rinse all over the surface of the flask, and remove the PBS after rinsing and dispose into a waste container. Add around 1 ml of trypsin to your flask and give it a nice swirl to spread around the surface area of the flask, which will help to detach the cells easily from the surface area if it spreads to all over the surface of the flask. So incubate the flask in the incubator for around 1 minute for the detachment of the cells. You can confirm the detachment of the cells by checking under the microscope, and if they're not detached completely, you can give a gentle tap at the bottom of the flask. And once they have completely detached, bring the flask into the hood back and add 0.5 ml of trypsin from the flask that you have incubated into a new flask, so that you spread the cells into 2 flasks equally. And after that, you have to add 3.5 ml of media to each flask and move the flask gently so that the media spread all around the surface area of the flask. And then you have to mark your flask with the passage number and your type of the cells and the date and the initials of your name if you want, and you can put back in them in the incubator and allow them to grow. And once after the cells reaching 80% of confluency, and you can passage them again. Usually the reaching of confluency depends on the type of your cells. Cancer cells grow faster than other kind of cells, so based on your cell type, you have to passage them again once after they're reaching 80% of confluency.